Myself Kumar from Siddipet, TSRJC. Let us now we are talking about the MGM structure. So MGM, so it is the MGM is the very important chapter for understanding the furtherly chapter photosynthesis and the respiration data. So in this chapter we can study about the MGM action and MGM structure, and MGM is affected by the different kinds of the factors, and the MGM classification and nomenclature we can study better in this chapter. So let us now we are understand the MGM chapter better. Already you have to the some knowledge. So for the enzymes. So when we are taking the food material to for the energy required purpose, we are taking the food. This food is digested in the stomach. So that's time this food is digested by the secretions of the some enzymes from the mouth to end of the part. Here better you understand the mouth. So for example, when we are taking to the food material inside the mouth, so the salivary glands are secretes the some enzymes like ptyalin enzymes, amylase enzymes. So these enzymes is reacted onto the like the substances. Amylase enzyme is reacted and gives to the glucose and converted into the maltose like substances. So here the definition is the enzymes definition is better. Here you are observing enzymes are catalysts which promote biochemical reactions in living organisms. Here you understand the particular catalyst that is the very important beta here. So that is the catalyst. What is the so sir? So here MGM catalyst. So the catalyst means the speed of the reaction, and the another one is the speed of the reaction, a rate of reactions. Sir, uh, screen share. Sir, screen share is stopped, sir. Okay.
Kumar sir, now you share the link.
Sir, okay. Okay. Now the MGMs is the main purpose is the what is that the MGMs shows the catalyst. Catalyst means what is the speed of the reaction, reaction and never change the equilibrium of the reaction beta. That is the MGM activity, MGM specific nature. So about the now we are discussing about the discovery of the MGM beta. Here you are observing Louis Pasteur. He is the very well known person in the MGM's chapter beta. Here you are observing Louis Pasteur. So while he conducted the one experiment, he find out the one micro certain microbes in the fermentation reactions. So those are called as ferments. So these ferments help into the fermentation of grape juice into alcohol. So Louis Pasteur, first scientist for the chance to the discovery and understanding the characteristic of the enzymes. So he is the first one person in the enzymes chapter. So then beta here you are observing enzyme. The enzyme term was coined by the W. Pune scientist. So actually enzyme is a Greek word. The enzyme name is enzyme means, so it is the yeast. So next one scientist here, the term gymase was coined by the Edward Buchner, the scientist. So he is isolated the enzyme from the yeast. So that's why he given name as gymase. So here beta another one scientist is there. So that is the next one, next slide. James Sumner. So James Sumner scientist is isolated a urease enzyme from the jug bean, and he postulated that all enzymes are proteins. So here beta you understand the he stated what is that all enzymes are proteins, but all proteins are not a enzyme. This is the very important point. So all enzymes are proteins, but all proteins are not the enzymes. So here, these four scientists are play the important role for the discovery of the enzymes. Now, only the proteins are there is the no chance to the only proteins are act as the enzymes, but some another another one chances is also there. So these are the, for example, the nucleic acid. So some nucleic acid also they behave like a enzymes. For example, a ribosome enzyme. So these ribosome enzyme is so derived from the 23S rRNA. So in prokaryotes. So in a prokaryote, it is a very important ribosome derived from the rRNA prokaryotes. Next one, eukaryotes also there. So in eukaryotes, the ribosome enzyme is derived from the 28S rRNA. So here you are observing the uh, ribosome enzymes are derived from the two types of the rRNA. 23S rRNA in a is the very important point to beta. So what is that here? Only proteins. Generally, we know what is that. All proteins are enzymes. So enzymes are all enzymes are proteins, but all proteins are not enzymes. But here, some nucleic acid also behave like the enzyme. So example is what is the beta here? Ribosome. The ribosome is derived the from the 23S rRNA in prokaryote. So ribosome enzyme is derived from the 28S rRNA. Okay, beta. Next one here you are having enzyme structure. So here you understand the enzyme structure is so it contain the a specific site for the fitting the substrate. So what is that here you are observing this? So rotating the area. So this is the every enzyme contain the a specific active site. So these active site means it is the shows the rigid and it is a sac-like structure. So these in these the substrate can fitted here, and then active substrate if it associated with the active site, and the active site can react on the substrate. So this is the generally the enzyme is what is that contain the specific site that is called as active site for the fitting the substrate. So this is the enzyme structure. So each enzyme is contain the what is that a particular site, particular sac like structure for the attached to the substratum. So this is the enzyme structure beta. Here you are observing generally in a first year we know the protein structure. So here enzymes, all enzymes are proteins. So that's why I'm telling that in a first year in a biomolecular chapter already we have to the some knowledge. So primary protein and secondary protein and tertiary protein. So these are proteins are there. Generally the tertiary protein is just like you are observing, it shows the number of folds and it appear like a woolen ball like structure. So it may be act as a protein. So in these generally four types of protein there. So from these four types only tertiary protein can be act as a enzymes beta. So that is the very important to beta. So what is that? Only tertiary protein can be act as a enzyme. So the these having to the what is that crevices or the packets to the involve into the formation of the enzyme. So these are the tertiary proteins beta. Tertiary proteins importance is what is the beta? The tertiary protein is shows the woolen bowl like structure. It may be act as a enzyme. So it consists of the what is that a crevices or packet like structure for the formation of the enzyme. So it is the very important point to beta. So next one tertiary, it is the what is the this structure is tertiary structure of protein beta. So next one. Now we discuss about the chemical reaction. So here the chemical compounds changes are two types beta here. So what is that? Chemical compounds are changes to two types. So that is the one is you are observing The chemical compounds or changes are two types beta. That is the very easy to understand these two types beta. Here already you know very well understand in chemistry subject physical changes beta first one. So physical changes, and second one is the chemical changes beta here. So first one physical changes. So physical changes means what is that? There is the no chance to the broken the or break down the bonds, but only the physical appearance can be changed. For example, ice is melt automatically, they can convert it into water, and water is given to the more temperature, automatically it was converted into vapor. So all are water, H2O, but the physical changes is there, water, ice and vapor. These are the changes. These changes is called as what is that physical changes beta. So here next one important is chemical changes. So in a chemical changes, so the bonds are broken and new bonds are formed during the transformation, called a chemical reaction. So here you take the, we are take the one example beta. Here you are observing barium hydroxide. Here you are observing barium hydroxide. Here you are observing H2SO4. Here there H2SO4, and here formed the what is the beta, barium sulfate, and the water here formed. So here carefully you are observing the barium hydroxide is, it is the one substrate or it is the one compound, and another one is the what is that H2SO4 here. So the barium hydroxide is the bonds between the barium and hydroxide. These bonds are cleavaged, and here H2SO4. So here also bond cleavage. So these barium is combined with the, here you see beta. So barium is combined with the sulfur beta. Here we can get the product as a what is that, barium sulfate beta, and release the water molecule. So here you understand what is that, here in a chemical reaction, actually here what is that, barium hydroxide beta, and it is the sulfuric acid. Now we can get the what is that, barium sulfate. It is the another one structure. So how it is possible to the form the a new product? It is the new product means what is that, this barium and sulfur. Between the barium and sulfur, the bonds are formed between these. So that's why we are getting the barium sulfate beta. So next one you are observing the rate of a physical or chemical process refers to the amount of product formed per unit time. Here you clear observe beta. Here rate is also known as velocity. Rate is also known as velocity. So here how much products are released, how much products, here you are observing how much products are released per unit time is called as, so unit time is called as a rate beta. So if you are take an example here beta, above example you see. So barium hydroxide, sulfuric acid, these are and here the sulfate here. So sulfate means is how much products. Here what is the how much products? So here sulfate is how much products are formed within a time. So per unit time, that is called as rate or velocity beta. So this is called as rate or velocity. So next one we discuss about catalysis reaction. So these reaction can gives to the information about the presence of enzyme, the it is it we can discuss about here better. So just I am taking the one example. So he understand the carbon dioxide is there and combined with the water. Yes, so carbon dioxide and water, so both are combined and gives rise to the carbonic acid. So the carbonic acid is what is the lemon soda. So these can be used into the preparation of lemon soda better. So here we observe that. So next, in a carbon dioxide and water molecule formed the what is that, carbonic acid. So without the presence of the enzyme, for example in this reaction is, so what is that, carbonic anhydrase there. So here you are observing carbonic anhydrase is there. So in the absence of carbonic anhydrase, uh, carbonic anhydrase. So, 200 molecules are formed per hour in the presence of enzyme. So, if the I am saying what is the beta, without presence of enzyme, carbonic anhydrase is absent. That's time the carbon dioxide and water molecules can give the carbonic acid. So, 200 molecules per hour. In the presence of enzyme, the carbonic anhydrase, for example, presence of enzyme, that's time the carbon dioxide combine with the water and gives rise to the carbonic acid 6 lakhs molecule per second. So here understand better. So without presence of enzyme, that's time we can get the only 200 molecules per 200 molecule per hour better. At the present of carbonic anhydrase enzyme, that's time we can get the 6 lakh molecules per second. For hour 200 get, in the presence of enzyme, we get the 6 lakh molecules per second beta. So what we understand here from these reaction, in the presence of enzyme, the reaction can very fast, and absence of enzymes, the reaction can very slow, and the product will be released very low quantity. So next one here, discuss about the beta metabolic and give it. So already you know beta glucose is six carbon molecules, and these are converted into the 3 carbons of 2 molecules of the pyruvic acid. These can be, we can study it in the glycolysis beta. The glucose, the 6 carbons, 1 molecule of glucose is converted into 2 molecules of pyruvic acid. In these reaction between the 10 reactions, glucose formed as a pyruvic acid. In between the 10 reactions are there, 10 enzymes are there, multi enzymes are involved for the formation of pyruvic acid here. So here, these pyruvic acid again involved into dehydrogenation and decarboxylation furtherly, and it can be gives rise to the acetyl enzyme active. So these acetyl coenzyme A is generally the acetyl coenzyme is act as a linker enzyme between the glycolysis and Krebs. So in a metabolic pathway, so for the formation of a new substances, between the formation of new substances involved a number of complex enzymes. So that is called as metabolic pathway. So here another one we take the situation, chemical conversion. So here beta chemical conversion means, so here the enzyme is act on where beta substrate, and when the enzyme is reacted on the substrate of beta, so then these substrate as a product. So here the what is that, the substrate can convert it into product. So how it is possible? Here you understand the what is beta. It is the enzyme. I am taking the diagram here. You observe this. The enzyme is contain the specific active site beta, and in these the substrate the resembles the substrate structure, and the enzyme active sites are the resemble both are. So these are fitted in the enzymes of active site. Then here formed. These are the what is beta. It is the enzyme, and here what is that fitted inside the active site of enzyme. So it is the what is that substrate beta. So here you clear observe that beta enzyme and the substrate. These are here formed the what is the beta enzyme and substrate complex. These are called as enzyme substrate complex. So this is the, it is not a permanent stages. So it is the afterward, after formation of enzyme substrate complex, then after the substrate can converted into the product beta. So that's why enzyme substrate complex is not a permanent stages. So here concept of activation energy we can discuss beta here. So here you observe beta, it is the best example also there. So in this we are taken the another one example. The x-axis is represent the progress of reaction beta, and the next one. So another one is also y-axis represent the beta here, potential energy. So that's why here the draw is observed here, the transition state. So these the dot lines are indicated the what is that substrate. So in this you are observing. So previously we discussed about the what is that pyruvic acid gives to the what is that like glucose to pyruvic acid in this reaction. So in a metabolic pathway we can understand the number of examples also there. In yeast involved into the form of the fermentation. In fermentation we can get the alcohol. These like substances, more enzymes are involved in that. So these are the metabolic pathway. So some are also there in the presence aerobic anaerobic condition form of the lactic acid. So these like substances are formed, different kinds of the substances are formed in muscles. So in a aerobic conditions, anaerobic conditions, lactic acid formation. So these are pyruvic acid formation. These are the we are taking the example for the metabolic pathway. Now we are discussing about the concept of activation energy. So here we what is that I am saying here, what is that in a x-axis represent the progress of reaction and y-axis the potential energy. So these dots are indicated the what is substrate. Here you are observing. So active energy means any substrate can the required the energy for the involvement of the reaction, and it can be help into the conversion of the product. So the substrate can need or required for the involvement into the reaction and for the conversion as a product. So here energy is required. That required energy is called. So some free energy is required to initiate, which is the conversion of the substratum. So that is called as activation energy beta. Here we take the examples very. So without enzyme, the substrate can require the more energy and reaches to the maximum state. This state is called as transition state beta. So without enzyme, that stem required the more energy, and after this enzyme substrate complex, and this is not a permanent stage. Here enzyme, here you are observing. Here the is a transition state, enzyme and substrate complexes are formed, but here not a, it is not a permanent stage. Then immediately they converted into as a product. The substrate can converted into product. So without MJ here very require the more energy. So that is called as activation energy is here required more energy. This is this line is indicated the more energy without presence of MJ. And next one you are observing here this blue color line beta. So here the in the presence of MJ, so there is the no need of there is the no required the high energy for the conversion of the products from the substrate. In the presence of enzyme there is the no required the more energy. So that's why it is indicated the what is that. So without enzyme required the more energy, more energy. In the presence of enzyme, so that's time there is the no required the high energy. So this is the concept of activation energy beta. So simply you learn here. Without MGM, that stem require the more energy. Okay. With the MGM, there is the no need the high energy. So that is the concept of activation energy beta. So here, this is the explanation here also. If P means what is that product is lower level than substrate, it is an exothermic. If the what is that lower products is formed lowerly than the substrate, that's time it is indicated the exothermic. Exothermic means what is that? Exothermic releases the some energy to outside, that is called as exothermic reaction. And next one, so the differences between the average energy content of substratum and that of transition state is called as activation energy. Already we completed this. So MGM eventually bring down this energy barrier, making the transition of substratum to product more easy better here. So this is the, in this concept we understand what is that activation energy is required more in the absence of enzyme. That is the very important concept in this. So next one, we are discussing about the mechanism of enzyme action better. So here, nature of enzyme. Here first we take the just diagrammatically I'm explaining here. So first, here you observe enzyme is there. So the enzyme is combined with the substratum. So the enzyme is, what is that? The enzyme contain the, what is that, active sites, and these active sites are the substratum is fixed in the active site, then bind closely bind to the substratum, then here form the, what is that, enzyme substratum complex beta. It is not a permanent stage. After formation of enzyme substratum complex, so immediately the substratum can converted into product beta here. So here you are observing these, first one, what is that, enzyme, and fitted the active side the substratum, then formed the, what is that, enzyme substratum complex. Then immediately these substratums, so it is not a permanent stage. Which one? So enzyme substratum is not a permanent stage. Then immediately the enzyme react on the substratum. Immediately the substratum can convert it into the products. So that's time after formation of product. So the product is released and enzyme also released as easily. So these enzyme is again combined with the another one substrate. So this is the, these action is called as, what is that, mechanism of enzyme action. So in here very important beta, you understand what is that. First, enzyme is contain the specific active site, and the substratum is fitted inside the active site and formed the enzyme substrate complex beta. And these substrate can it is not a permanent stages. Immediately the substrate can react with the enzyme, and the substrate can change it as a product. So that's time again they are the separated enzyme and products. So this is the mechanism of enzyme action. So for understanding purpose, so the mechanism of enzyme action is, so two theory are there. So very important theories are there. So that is the first one theory. So one is the lock and key hypothesis. So this is theory is a very popular theory beta. So it is a very important for the examination also, IP examination also. You understand here very easy. So lock and key hypothesis beta. So it is proposed by Emil Fischer, scientist beta. So I am taking the one diagram for the easy understanding. So here you are observing this diagram beta. So in this diagram you are observing here. So enzyme already we now recently discussed here the enzyme molecule and the substratum. The substratum are fitted in the enzyme active site. So and here formed the, what is the beta, enzyme substrate complex are here formed. You, so these substratum are fitted in inside the active site of the enzyme beta. So it is the, what is that, enzyme substratum complex. Then after these product, these substratums are converted into product here. Then after the enzyme is released from here. So it is we are comparing with the lock and key. Means here the substratum is imaged as, what is that, it is a lock, and key is imaged as a enzyme. So here you are observing. So these key, the enzymes active sites resemble with the substratum lock structures. So when the key is inserted in the substratum means lock, then that is the, when we are key inserted in the lock, that is called as, what is that, enzyme substrate complex. When we are rotate in the substratum, the keys we are keys rotate in the lock, inside the lock, immediately the lock can be open. So the open is called as, what is that, product. So here we understand what is that, our house key is fitted in the our house lock only, but our key cannot fitted to the beside house So that's time this key is not open the another or beside house of the lock. So here we understand what is that a specific key can react with the specific lock. So these MGM cannot react with another one lock. So here this theory can be explained to us. So what is that? It can be explained about the engine specificity better. So means what is that? A specific engine can react on the a specific substratum. So this is the very popular theory beta. It is the lock and key hypothesis. Understand better? So this theory is the very popular and acceptable theory. So for the understanding the engine specificity, each engine is contain the a specific specificity. Specificity means what is that? The engine is a react on a specific substratum. So this theory is explained about the what is that enzyme specificity better. So understand better. It is the very important. First one, what is that? A key. Key is imaged as what is that enzyme. So substratum imaged as log. A specific key can react with the a specific log, and then log will be opened. So it can be explained as what is that engine specificity. A specific key can open the specific log. So this is the very important beta lock and key hypothesis. It is the very important for examination purpose beta. Next one, another one here also beta, induced feed hypothesis. So it is not a acceptable theory, it is utter failure theory beta. Why? Because means it is proposed by the Koshal and beta. So in this same similar, so you are observing mgen consists of the active sites and the combined with the substrate beta. So here one is different. So the substrates are in this according to this theory, the specific substrates are not fitted in the specific MJ. So, but he explained what is that another one substrate, any another one, any kinds of the substrates involved in the different kinds of the engines, but it is not a possible. So, if possible, the engine substrate complex is formed is possible, but the engine cannot react on the different kinds of the substrate. But he explained about the various engines can react with the various substratum. It cannot be explained about the engine specificity better. So here you are observing better. It is a utter failure theory better. So in this you are observing. For example, I am taking the example enzyme is, for example, it is urease. We think that urease. Actually, urease can react on where better urea. But here, according to this theory, this is the sucrose. So this theory explained what is that this is the urease enzyme and this is the sucrose. So the sucrose substances fitted in the enzyme. Enzyme which is which enzyme? It is the sucrose enzyme. So sucrose enzyme is react on the which one? Another one substances. So it is the maltose enzyme. It is the sucrose. Another one, it is not possible to the react on the sucrose. Generally, urease enzyme can react on the urea, but not the sucrose. So he explained about what is that the urease enzyme can react on the sucrose. So it is possible, better. It is not possible. It cannot explain about the enzyme specificity. So that's why this theory cannot explain about the enzyme specificity better. So that's why it is a utter failure theory. So about the enzyme reaction, very important. First you should write the enzyme substrate complex, enzyme substrate complex bind. Then after the enzyme can cleavage the bond of the substrate, and the substrates can converted into product with the broken of the bonds. So in these two theory we learn better. One is the lock and key theory is completed. Then after induced feed hypothesis also completed. But in these two theories, only acceptable theory is what is that better? Lock and key theory better. Induced feed hypothesis is utterly failure. It is not acceptable theory because of it cannot explain about the enzyme specificity better. So next one, we discuss about the uh,
Screen share ಆಯ್ತು ಸ್ಕ್ರೀನ್ ಶೇರ್ ಆಯ್ತು ಸರ್
Kumar sir,
Okay, sir.
close
Yes,
the
please.
session.
Sir?
Now you shared.
Okay, just wait, sir.
Remaining for tomorrow.
Okay, better. Now we discuss about the factors affecting MGM activity better. So factors affecting MGM activity in this first factor is the temperature. So in a temperature, so low temperature preserve the enzymes in a temporarily inactive state, whereas high temperature destroy the enzymatic activity because proteins are denatured by heat. So actually here you observe better, optimum temperature is 40 degrees centigrade better, 40 degree centigrade. Above the 40 degrees centigrade, the enzymes are inactive. So the optimum temperature uh, active when the optimum temperature is uh, sorry here first one, temperature. So okay, temperature is above the optimum temperature can affected to the inactive the enzymes beta. So today we completed the enzyme action. Tomorrow we will again discuss about the factors affecting enzymes activity beta. Okay, thank you all of you. Bye.